Hello everybody, welcome to the Riots Club, today at 6 p.m. exceptionally. So, of course, it's not the lunch date that we usually have, but today we have a special guest from California, and time difference makes it so that it's a, let's say, pint talk today. So before we start, just to show you the next talks, just to remind you, so next week we have Olivia Guest, and then Emily Sina, Lisa Lynn, Fiona Fiddler, then Quintana, and we're working on the rest for the rest of the summer. Should be quite exciting, actually. And also, don't forget, on the 8th of July, we have this amazing event that should be coming. The Cost of Correcting Bad Science, organized by the Riot and Reproducibility. It should be amazing, guys, so please do subscribe on Eventbrite. Now, it's with great pleasure that I'm going to introduce Professor Russell Poldrack. I'm quite nervous, to be honest, because I've been watching his work since the beginning of my work. Been working with fMRI for a long time, working with inhibition, gonago, step task, all this. So I've been following his work for a long time, and it's a privilege to have him here today with us. So Professor Poldrack is the Albert Ray Lang Professor in the Department of Psychology and Professor of Computer Science at Stanford University and Director of the Stanford Center for Reproducible Neuroscience. I mean, we have someone who's the director of a center that is for reproducible neuroscience. Who best to represent this talk today, right? His work is mainly on neuroimaging and how to understand brain systems underlying decision-making and executive functions. His lab is also engaged in the development of neuroinformatics tools to help improve the reproducibility and transparency of neuroscience, including Open Neuro NeuroVault, data sharing projects, and the Cognitive Atlas Ontology. I'm very pleased to welcome you today, Russ. Please.
All right, well, thanks very much for the invitation. It's a pleasure to be here and talk about reproducibility, and thank you for changing the time so I didn't have to get up too early this morning. So first, I just want to say, what do I mean when I talk about reproducibility and reproducible science? And I think, there's lots of different things this can mean. And a paper by Steve Goodman and colleagues a couple of years ago kind of nicely laid out sort of the different ways of thinking about this. Everything from, what a lot of the work that my lab has done has been around what they call methods reproducibility, which is basically, can you repeat the analyses and the study, based on the description. Most of the field, I think, has been largely focused on what they call results reproducibility, or what, how can you replicate particular results? And there's also other ways of thinking about it that are kind of more general. I'm going to focus really on, most of the talk will be focused on methods reproducibility, in part because I think, thinking about results reproducibility in the context of imaging is challenging, right? So we saw a few years ago this paper in Psychology that tried to, replicate 100 published findings from several journals and found that they could only replicate about a third of those in terms of, finding statistically significant results. The challenge would be that if we wanted to go try to replicate 100 published imaging studies, that would be a really expensive effort. And so the question is, is there any other things we can look at to know whether our results are reproducible? And one is just to look at meta-analyses. So here's this is a figure from a meta-analysis published a few years ago by Mueller et al. Of looking at brain dysfunction and major depression. So they took several previous meta-analyses. And so they're showing actually each of the dots here is a region that had been found to show a significant difference between healthy and depressed individuals in a meta-analysis. And they re-meta-analyzed the 99 studies that were sort of, underneath all this, and basically found that when they did the meta-analysis correctly, they fixed some of the issues with previous meta-analyses. They found no consistent significant differences between healthy and depressed individuals, even though each of the individual 99 studies had claimed to find something, and each of the previous meta-analyses had claimed to find something. Similarly, if people have looked at correlations between brain and behavior, and this is a paper published by Fowler-Buchel several years ago that tried to replicate a number of different correlations between brain and between structural imaging and behavior, and basically found that none of them were really replicated. And so this suggests that that there's a big problem in terms of, particularly I think in the context of things like group differences and individual differences, where the studies have, as we'll see, largely been way underpowered. So this is a mess, right? And so we could kind of stick our head in the sand, or we could go do something else. We decided to try to fix it. And so with funding from the Arnold Foundation a few years ago, we started a center called the Stanford Center for Reproducible Neuroscience, with the goal of trying to improve science, improve the reproducibility of science, at least in the area that we work in. And the way that I think about this is in terms of a design problem, right? So if I was in the room with you physically, I would ask you to raise your hand if you knew what this thing was. And some of you might know that this is a commercial paper cutter, and it has 2 green buttons on it. And those green buttons are basically there because, if you don't make humans do something with their hands, they're likely to try to hold the paper in the cutter and end up cutting off their fingers. And so the designers thought about the stupid things that humans would do and designed the machine to prevent them from doing that. And that, in fact, is in general one of the principles of design. Figure out what you want people to do and what you don't want people to do, and design the artifacts in order to afford the things you want. Now, in the context of something like fMRI analysis, we can think of design in terms of what's called choice architecture by behavioral economists. This book by Thaler and Sunstein sort of laid out this idea of nudges. The idea of choice architecture is anytime we go into a situation where we have to, make particular choices, we're going to be biased towards some choices and away from some other choices. So, for example, if our software provides us with a default threshold, we're very likely to use that default threshold. So nudges are the idea that we can sort of improve behavior by changing the choice architecture without limiting anyone's freedom. You can always still choose the whatever threshold you want, but we want to sort of use defaults, provide feedback, expect errors, and prevent errors in a way that helps sort of improve behavior. So what are we doing in our center to try to help? We focused around several different areas, and I'll kind of cover some of each of these in more or less detail today. One is open data. We've worked on a number of projects that are trying to provide people with really open access to data at various levels. Another is open tools. Some of the tools that we focused on are like quality control tools like MRI-QC, pre-processing tools like fMRI prep, and tools to improve power and design. A lot of this has relied on open standards, particularly the brain imaging data structure, or BID standard, that we've put a lot of effort into in the last few years. And finally, I'm going to talk about some work that we've done in the context of what we call meta science, which is basically trying to understand how science works using science. And if you're not familiar with this paper, I'm sure if you're part of this group, almost all of you must have read this paper, Why Most Published Research Findings Are False, which really kind of, I think, was the first shot in the reproducibility crisis by John Ioannidis. And so John laid out a number of features of a scientific field that he said were were likely to cause findings to be true or false. And one feature he noted was the smaller the studies, the less likely the findings are to be true. The greater the number and lesser selection of tested relationships, the less likely the findings are to be true. And the greater the flexibility in designs, definitions, and analytical modes, the less likely the findings are to be true. So first, let's focus in on this one, the smaller the studies. And since this paper a few years ago by Kate Button and colleagues, it's been really clear that neuroscience in general has a statistical power problem. So remember, statistical power is the likelihood of finding an effect if it's actually there. When they looked both at structural imaging studies and rodent studies, they found that the majority of studies were badly underpowered. Many of them had so little power. If your study has 10% power, that means that even if there's an effect there, 9 times out of 10 you're not going to find it. And that's that. So basically, that research is pretty much futile. But sort of going back to the arguments that Ioannidis made in his original paper, one might argue, in fact, people, when I've showed that previous slide have argued, well, that's fine. As long as we control our type 1 error, then we don't have to worry because, we're only going to have a specific number of false or specific proportion of false positives. And that's true, but that isn't necessarily the rate that the number that we care about. And Ioannidis argued that what we really care about is the positive predictive value. That is the likelihood that when we get a positive result, that it is true, right? So instead of conditionalizing on all tests, we conditionalize on only the positive outcomes. And then the question is, of all of our positive outcomes, how many of them are true? So here's a thought experiment. So let's say we're a physicist and we're doing, some experiments, but our detector is broken. So it's just giving us white noise. And so, if we do 100 studies, we're going to get 5 significant results, simply due to chance if we set our alpha at 0.05. But all of those are false positives, right? Because there is no true positive. It's all just white noise. So our positive predictive value is 0. Right? And so what Ioannidis showed was that basically, as your power goes up, the positive predictive value goes up, right? The likelihood of positive findings being true positives goes up. And for underpowered studies, even if you find a positive result, it may be, highly unlikely that it's actually a true finding. They also showed that they also discussed the fact that, if you're underpowered, any positive finding you have heavily overestimates the true effect size. So if we want to ask about power in fMRI, the one challenge is knowing what a realistic effect size is. We can't take published effect sizes because they've come from analyses where, where you've already done some kind of selection. So they're going to necessarily, if you've thresholded and only taken the significant clusters, then estimating anything about those clusters is necessarily going to be an overestimate. And so in this Nature Reviews Neuroscience paper published several years ago, we took the Human Connectome Project data and tried to estimate realistic effect sizes by basically, taking the data, getting a group map, and then using the regions of interest that had been published by the Human Connectome Project investigators, along with maps from Neurosynth, to try to basically focus in and find where are the voxels that we are really certain should activate, for example, for a motor task. And then we asked, what's the effect size within that region? And that gives us an unbiased effect size estimate. So when we do that, on the left, you see the percent signal change. That's kind of the size of the within subject effect. And this is just to show that these are all whopping effects, right? These are all in the order of like 2 to 3, up to 4% signal change, which is an order of magnitude bigger than what we would expect for kind of an interesting cognitive phenomena, because these are almost all just like task versus baseline sort of contrast. But that's not the effect size we care about, because that isn't with respect to the variability across subjects, right? So we also computed Cohen's d, which basically says how big is the effect with respect to the variability across people. And what we see there is that, most of the effects are in the kind of medium range in terms of Cohen's d. The biggest one, for example, in supplementary motor cortex for a motor task is around, 0.7. Some of them are down, closer to say 0.2. Those are all like, kind of decent effect sizes, but certainly not whopping. So then we can ask, how well are fMRI studies powered to find these kinds of effects? And this is, these sample sizes are a bit out of date now, right? And certainly things have gotten a little bit better since then. But if you look back, up to 2015, in 2015, the median study was powered to find activation with effect size about 0.75. What that means is that half of the studies published in 2015 were not even well powered to find kind of the biggest effect you would ever expect. So they're badly underpowered to find sort of an interesting effect. Now, we've also seen since then that, that this extends to work in using machine learning tools as well. This is a really nice paper by Gael Varacqua from a few years ago, which basically showed that across lots of different phenotypes, as sample sizes get bigger, predictive accuracy of machine learning tools goes down, right? And that's fully expected by, if you have any sort of publication bias, people are going to, the estimates of predictive accuracy are much more variable for small samples than for large ones. And if you're only taking the ones that look good, even without any sort of, accuracy hacking, if you're only taking the ones that look good, you're going to have an overestimate, a bias towards larger effects for larger accuracies for smaller samples. Now, I think one of the challenges here is, and I assume that, many of the people on this seminar are early career researchers. And, one of the big challenges is that what does this tell us? This tells us that we need big samples, right? But many early career researchers just don't have the resources to go do the kind of sufficiently powered study that you need to do. So I wrote a paper in Neuron last year that kind of talked through some of these issues and talked about like what early career researchers can do, because I don't think the answer is to go do an underpowered study and just pretend it's okay. And I think that Nancy Canwisher had a great quote: "If you can't answer the question you love the question you can." And I talked in that paper about various things you can do: collaborating, using shared data, focusing on theory. And in our lab, we've really been focusing a lot on the use of shared data. I've had postdocs who came and spent 2 years with me, got a bunch of papers only using shared data. And so, if you're interested in shared data, the Open Neuro repository that we run now has a large number of data sets across multiple species, lots of different tasks, different types of data, and extending out beyond MR to MEG, EEG, and intracranial measurements. So I definitely recommend, if you're interested in kind of seeing what can you do with shared data, the Open Neuro is a platform you should take a look at. All the data are shared completely openly, so you can download them without any sort of extra permissions or anything like that. So I want to turn now to the importance of measurement, right? So what makes a good measurement, right? And in psychology, we talk about, in measurement theory in general, people talk about two different aspects of measurement that are important. One is what we call reliability. That is, whatever it is that we're measuring, how precisely are we measuring that thing? So, as shown on the right, you can have an unreliable measurement that is on average like, hitting the bullseye, but it's kind of all over the place. A much more reliable measurement, like the one on the bottom, hits the bullseye much more accurately with much less variability. Now, this is separate from the question of validity, which is, are we measuring what we think we're measuring, right? It could be that we have a really precise measurement, take COVID-19, right? It could be that we have a measurement that is really measuring H1N1 flu rather than COVID-19, but it's highly reliable at measuring H1N1 flu. It's a reliable measurement, but not a valid measurement of COVID. So this became important in the imaging literature a few years ago with a paper that many of you may be familiar with. It was initially titled Voodoo Correlations in Social Neuroscience, and it was retitled to this long, weird title. And they pointed out that they were seeing a ton of papers in the literature where people were publishing these what seemed like really like way too high correlations between brain and behavior. And they pointed out one of the fundamental aspects of measurement theory, which is that the correlation between two measures is bounded by the reliabilities of those measures, particularly by the geometric mean of the reliability. So here's an example of one of those puzzlingly high correlations. This is a paper published back in 2007. This wasn't one of the ones that they talked about, because this is more from sort of neuroeconomics, where the investigators found this correlation in a small sample between a behavioral measure of loss aversion and a neural measure of loss aversion, and reported this. And obviously it was very impressive because it got into Science. That investigator happened to be me and my group, and so I'm not trying to throw shade on anybody else other than myself here. When I saw this, when I saw Bull's paper and realized, hey, what we've been doing was really dumb, we spent a lot of time trying to figure out, one, how do we fix this, and also, what does it mean? So Jeanette Mumford and I actually wrote a follow-up paper that sort of dug a lot more into this. The point is that we need to care about the reliability of our measurements, right? This is just kind of laying out what I said about how a variable can only correlate with another variable as strongly as it correlates with itself. You think about reliability as basically how well does something correlate with itself. Something can't correlate with anything else better than it correlates with itself. So what do we know about the reliability of fMRI? Well, it turns out to be pretty bad, and this is shown just recently in a paper by Max Elliott and colleagues published in Psych Science, where they looked across both across two large data sets, the Human Connectome Project and the Dunedin study, and show that for a bunch of these different measures, reliability never exceeds 0.5, right? So in general, anytime somebody tells you that something is correlating that an fMRI measure is correlating with anything else higher than 0.5, you should probably be worried about that, about how that measurement came about. They also did a meta-analysis of lots of studies of reliability from the literature and showed that the average reliability, the average test-retest reliability was about 0.4. So this says that we need to worry a lot about reliability. Now, what do we know about reliability? Well, it turns out that there's a lawful relationship between reliability and the amount of data in general. It's called the Spearman-Brown prophecy formula. And so, for example, when in one of the studies that we published that came out of the MyConnectome project, where I scanned myself repeatedly over the course of a year and a half, we looked at the reliability of connectivity estimates from resting fMRI by doing sort of split half correlation. And what on the top you see basically, a couple of different ways of looking at reliability that showed that reliability gets up into a reasonable range with about 30 minutes of data, kind of maxes out around 100 minutes of data. And the bottom shows that basically we could predict the data, we could predict that reliability curve very well using just a small amount of data based on a version of this Spearman-Brown prophecy formula. So I think we need to, one of the things that we need to think about is like, how can we not just, improve our statistical power, but also improve the reliability of our measurements? And clearly, collecting more data per individual has become, I think, an important aspect of that. Okay, so now let's turn to another aspect of data analysis in neuroimaging, which also relates to some of the points that Ioannidis made. So he said the greater the number of and lesser selection of tested relationships and the greater the flexibility in data analysis, the more likely the findings are to be false. So this is from that same study of the same review paper we published a few years ago in Nature Reviews Neuroscience, showing this is just for FSL, looking at the number of possible workflows that one could generate just using different choices at different steps in the process. And what you see is that using reasonable steps, there was on the order of more than 69,000 possible workflows. And we know that these actually this variability in workflows does actually exist in the literature. So Josh Karp did this study a while ago, The Secret Lives of Experiments, where he just looked at how flexible, how many different types of workflows do people report using, and saw that there was a huge amount of variability. There was nearly as many unique analysis pipelines as there are studies in the sample. And these seem to make a difference, right? So he showed if you take one data set and analyze it with almost 7,000 different pipelines, you can get differences in the z-statistic values on the order of 8 standard deviations between the highest and the lowest. So that's a huge amount of variability in the statistical values that one could find. So I think one thing that this points out is the importance of pre-registration, right? Particularly around sample size and analysis plan. And I'm suggesting something here that we haven't actually done yet, which is that you actually pre-register your sample size and your plan with code that's based on analysis of simulated data, so you can show that everything works before you actually run it on your real data. Obviously, that's a ton of work up front. Important to point out that pre-registration doesn't prevent exploratory analysis. It mostly just helps you be sure what really was exploratory and what really was planned, as long as you're clear in the paper about what was exploratory and what was planned. Now, the effects of pre-registration in other fields have been really astounding. So this is from a review from a few years ago, looking at what happened when one of the NIH institutes that funds pharmaceutical and dietary interventions started requiring pre-registration. So in the year 2000, they started requiring clinical trials to register their primary outcomes. That is, what is the thing you are measuring your effect on? And so previously, so before 2000, the large majority of clinical trials reported finding a benefit of their treatment, right? These are the plus signs, and nobody reported finding a harmful effect of their treatment. go past 2000, and what you see is now suddenly nearly every trial is showing a null effect. 2 showed benefit, 1 showed harm, right? So this is basically saying when you prevent people from moving the goalpost, suddenly they're much less likely to kick a goal. Now, unfortunately, under the current incentives to publish positive results, this is a cost, right? This means you're hurting yourself by pre-registering, right? And so I think that's an important thing for us to think about how to change. Okay, so, and it's also important to point out that pre-registration will prevent p-hacking, but it doesn't eliminate analytic variability, right? You could have 100 labs pre-register 100 different workflows. And so the question still remains, one, how variable are workflows in the wild? And what's their effect on the inferences that people make? So we recently published a study led by Tom Schoenberg and Rotem Botmandik-Nazer, who's now in Tor Wager's lab, that tried to look at this, called the NARP study, the Neuroimaging Analysis, Replication, and Prediction Study. And these are just some of the individuals who were involved in that study. So what we did was we collected data on a task, the same task that it used in that science paper that I talked about earlier, in which people make decisions, monetary decisions about gambles. And we collected 108 subjects' data at Tel Aviv University. And then basically we recruited people to, I'm sure at least one person who's involved is probably sitting in the audience here. We basically recruited teams of people to analyze the data. And we basically said you can have up to 3 people on your team. You have to sign a non-disclosure agreement saying you won't tell anybody about your results until the embargo is lifted. And we offered co-authorship on the initial paper, and that's why the paper that just came out has almost 200 authors on it. We gave out the data and basically gave people about 3 months to give us back a set of answers on 9 hypothesis tests of the sort of, is there significant activation in some particular brain area for some particular feature of the experimental design? And we told them basically use whatever standard analysis workflow use in your lab. The only thing that we said was that the analysis had to be a whole brain analysis. And so we got back 70 teams sent us back results. I think we started with 80 something who we gave data to. So almost everybody gave us back data. And we also, in addition to having them say yes or no on the 9 hypotheses, we had them give us whole brain statistical maps both before and after thresholding. So we also had them each give us a detailed written description of their workflow. And what we saw was that none of the 2 teams used an identical workflow. And also interestingly, even with these like multi-page written descriptions that followed the best practices for reporting, it was impossible to tell exactly what they had done. We had to, sometimes ask for the actual, code and the scripts that were used. So here is the effect of analytical variability on outcomes. So what I'm showing you here is the proportion of teams for each of the hypotheses that said, yes, there is a significant effect, sorted by that proportion, right? So this is basically showing that for 3 of the hypotheses, pretty much everybody agreed that there's nothing there. I think 5% of teams said yes. For 1 of the hypotheses, pretty much every team said, yes, there is something there. For 5 of the hypotheses, basically people were kind of in the middle, right? Same data set, but getting substantially different answers from these different teams. And in fact, if you look across teams at the patterns of yes and no they gave across these 9 hypotheses, there were 33 different patterns across 70 teams. And for any hypothesis, there were at least 4 workflows that could give a positive result or a negative result. And so that's a pretty striking and slightly frightening amount of variability. This is what it looks like if you look at the thresholded maps. This is showing the proportion of teams at each voxel who reported a significant finding. This is for the best, the highest overlap map, and we see that for the best contrast, the best voxel has 76% of teams saying yes, there is activation in this voxel. Now, interestingly, if we look at the underlying statistical maps, they're actually more similar than you might expect. So on the right, you see a heat map showing similarity of the underlying whole brain statistical maps. And what you see is that there's one big group that had actually fairly similar maps. But if you see along the top, there's red and green. Green means that they said yes for this hypothesis. Red means they said no. So you can see that even for teams that had pretty similar statistical maps, their yes/no decisions on the hypothesis could differ. Now you see that there's a second group, the group with the yellow to the left of it, whose maps were actually negatively correlated with the rest of the maps. And then there's a third group that were kind of uncorrelated with everybody else. So we looked more deeply into this second group, right, and found that they had all, well, for the ones where we could get the necessary information, they made model misspecification errors. There were two different errors. One was basically that they were supposed to build a parametric, it was supposed to be a parametric effect that they were testing, but they didn't remove the overall effect of task. And so what they're really looking at here is basically something that looks very much like just task activation. And it happened to be negatively correlated because the activation pattern for this particular task looks like a default mode sort of pattern. The second set of teams did something kind of more interesting where they added in basically a bunch of regressors that ended up messing up the interpretation of the gain regressor, causing it to actually look uncorrelated. And so that reflects this kind of general faith that people have that the general linear model is going to automatically fix collinearity problems, without realizing that it does fix it statistically, but it can change the interpretation of the resulting regressors. So we think that, a really important outcome of this is the need to do what goes by various names, what we've been calling multiverse analysis, which is basically running a data set through many different plausible analysis pathways, and asking, which particular results are convergent and consistent across those different pathways. Now, a challenge is, fMRI analyses are big, right? And so this is going to require, one, the ability to kind of automatically specify statistical models and then to execute those on high performance computing systems. So we've been working, we have a project called FitLens that Chris Markovich from my group has been developing that basically allows one to do this by using another part of the BID specification, a statistical model spec that allows you to basically write down a machine readable description of an fMRI model, and then FitLens can take that and apply it to a data set. So this allows us to create many different models and execute them all automatically. And we've just started working on trying to implement these kinds of analyses. Okay, so I'm going to turn now to another aspect of reproducibility, of particularly kind of methods reproducibility that we've become really interested in, concerned about, which is kind of software quality. So if I was in a physical room, I would ask you to raise hands about how many of you have written computer code in the course of your research, and pretty much everybody would. How many of you have been trained in software engineering? Quite a fewer people would raise their hand. How many of you have ever written a test for your code? Again, a fairly small number of people would raise their hand. And we know that software errors are a problem. And a really striking example comes from the work of Jeffrey Chang quite a few years ago. He had published several papers. He's a structural biologist, like a real hotshot. Had published several papers in Science, in addition to other papers that had looked at the structure of this particular protein. He had to retract those 3 Science papers along with a couple of other papers after it turned out that a piece of software they had gotten from someone else basically made one sign flip, and that completely changed the structure of the protein. And after somebody else realized this, I think they dealt with it properly. They retracted the papers and he's gone on to have a really great career. But it just shows how important software quality control is. And so I wrote about this in a blog quite a while ago. I was doing some analyses of some classification analyses on the open neuro data set, trying to basically say, we have people doing one of 23 different tasks, and I classify what task they were doing. And so this is just a little snippet of code I wrote using scikit-learn to do that. And I got 93% accuracy on my classification problem. And chance is 1 over 23. So this is way, way above chance. It's not perfect. And if it had been 100%, I would have been sure something was wrong, right? But it was 93%. So I'm like, oh, wow, this is a great result. And then one of my postdocs was a computer scientist. I had him look over the code and review it. And he found an error. He's like, oh, you left out one index on these matrices. So you're actually classifying across subjects rather than across tasks. And when I reran it, now I get 53% accuracy, still way above chance. But I was about to submit this paper, and we would have submitted a paper that badly overestimated the accuracy of our results. Fortunately, we were able to catch it before we submitted it. But this highlights a real problem especially when we write custom code for data analysis, which is what I've come to call bug hacking, right? Which is that if a bug in our code confirms our predictions, we're much less likely to actually find it than if it disconfirms them, right? If it gives us a quote unquote bad result, then we're much more likely to try to find out what's wrong with our code. Whereas if it gives us a quote unquote good result, then we're likely to say, hey, we got a good result, let's go. So I wrote another blog post a while ago about this idea that whenever I call the principle of assumed error, which is whenever you find something that seems like a really good result, you should assume that it occurred due to an error in your code. And this is one way to protect yourself from this sort of bug hacking. Now, another way to do this is actually to test your software, right? And software testing is challenging. Because in part because a lot of, if you look at software engineering, a lot of the things that software engineers do is sort of testing at levels that probably aren't going to be helpful for scientists. So one of the things that we do is usually just write what we call smoke tests, which basically just make sure that everything will run without exploding. This is really useful during software development to make sure that you haven't introduced a problem. Another thing that can be really useful is sanity checks. Make sure that our values within reasonable ranges. If you're, for example, if you have age values in your analysis, make sure that all of your ages are within the range of whatever the minimum or the maximum age for your sample should be. If not, then it should let you know that. I'll talk about some other types of testing that one might do in a bit. Another thing that we've become much more attuned to is using many of the different tools that are available in software engineering for analyzing the quality of one's code. And one of these is a set of tools for what's called static analysis, which can, and I'm going to focus here on Python because that's the ecosystem that we work in. Python has particularly good tools for what I'm talking about. It's more challenging if you're working in R or MATLAB. The tools just aren't as good, but they do exist. So tools like these will, one, tell you are there any obvious errors in your code, and two, are you adhering to the style guidelines? And one of the reasons to adhere to style guidelines is it makes the code much easier for people to read, for other people. So one of my collaborators, I gave them some code to review. They gave it back to me and said, sorry, you need to make that style compliant first because I can't read it. And once I fixed it, once I've made it be compliant, then they said, oh, wow, now I can actually read the code. Another thing that I think is important is basically whenever you can use well-tested established libraries. So there's a tendency, in scientists and engineers to have this thing that people sometimes call the NIH or the not invented here effect, which is, even though there's great solutions, I need to grow my own, right? So the canonical example for me is, I need to write a new DICOM to NIFTY converter, right? Sometimes you do need to write a new thing, but often, rather than writing a new thing, especially if it's, if you're working in open source, you can take existing things and add what you need to those, right? And then you can actually contribute them back to the project and they can become something that other people use as well. I think it's also important to prefer to use libraries that really use good software engineering practices, scikit-learn being, I think, a great example of that. So I want to just in the last few minutes talk through an example, kind of a case study of how we did this in the NARPS project. So, the NARPS project, each of the teams gave us all of their results, and then we needed to put together an analysis pipeline to analyze those results. And this is kind of a schematic of that pipeline. So it involves shared data. We have the data that the teams gave us that was shared through Zenodo. It involves shared code, the analysis code shared on GitHub, and actually a shared platform, which was something that was created, a container created using Docker. And then ultimately that was all put together using continuous integration that I'll talk about and shared as a result again on Zenodo. So the first thing we do is share the data openly and we version the data, right? So we know exactly what data set was used by each of the teams for their analyses, right? And that data set actually has a DOI. We published a scientific data paper describing it. So you know exactly what went into each of the teams. And this was supported by the BIDS standard that I mentioned earlier, which basically allows us to give people data and have them kind of know immediately how to analyze it. And so I'm not going to say more about BIDS, but I think many of you have probably heard of it. If you haven't, it's check out biz.neuroimaging.io. We also versioned the data that we generated that were generated by the team. So every time we did an analysis run, we used a fixed version of the data that again had a DOI, was shared at Zenodo. So the goal here is to have a completely reproducible path from original data, in this case, the data that the teams gave us, up through the figures and the final numbers that go in the paper. Likewise, we versioned the code. So each version of the paper was associated with a fixed code release. So every time we said, hey, here's we're submitting a revision, we're sending this to the co-authors, we had a fixed code release that went into generating those results. Now, I mentioned the idea of containerization, and this part of the rationale for this comes from this quote by David Donahoe. An article about a computational result is advertising, not scholarship. The scholarship is the full software environment, code, and data that produce the results. And we know that different operating systems can lead to different results. And containers provide a way to share an entire computing platform, right, including in particular all the specific versions of the software and the operating system libraries that are being used. And we use Docker, which is a really handy tool for this. And so in Docker, you basically write a file that describes all the things that need to be sort of put together in this container, and then you can share that container and anybody can take it and reuse it. Now this allows us to then basically use a software engineering tool called continuous integration. So anytime I pushed new code to the GitHub repo for NARPS, it would automatically trigger Circle CI to grab all the data, grab the Docker image from Docker Hub, and then run all the analyses, rerun all the analyses on the data, and make sure that they run, and also do some tests to make sure that that things look right. And then basically the results from that, the artifacts that are generated, like the figures and things like that, are saved in a place that we can then download and share. Now we also, in addition to sort of code quality control tools, we also use human code review, and I think this is really important, to have more than one person looking at a piece of code. And I think this is not just useful for for kind of error checking, but also for helping people learn how to code. Like I learn something every time I do one of these code reviews, and we've started doing them regularly in the lab. And so the main goal was just to figure out, does my logic make sense? Am I doing the right thing? And also to look for kind of stupid mistakes that I might have made. And so for NARPS, Tristan Gotthard from Concordia kind of went through all the code and gave me back a bunch of comments and was really helpful. Finally, one of the things that's important to realize is that reproducibility doesn't mean that something is correct, right? A result can be fully reproducible but incorrect, and this really gets at the distinction between reliability and validity. And so one of the things you want to do is make sure that that it's actually giving you back what you expect. And so there's a couple of ways to do that. One is to basically generate some synthetic data for which the true answer, and then basically make sure that your code can recover that true answer. The second is to kind of the opposite, which is to generate data where where you basically have made the null hypothesis true. So basically generate noise data and make sure that your observed false positive rate is accurate. That when you do this on 1,000 noise data sets, you only get 50 positive results, or whatever your false positive rate should be. Obviously, it's best if you, as I mentioned earlier, if you do this in advance of any real analyses, because it really helps you kind of make your code bulletproof before it ever touches the real data. Again, this is something that we're just starting to do in my lab, but I'm hoping that we can make it a bigger part of what we do. So for NARPS, what we did was we generated simulated data for each analysis team, including some of the weird things that we saw across teams. And then we basically ran the entire analysis stream on the simulated data and compared the results of the final analysis to the ground truth maps that were used to generate the data, and made sure that those, in this case, we basically said, if everything correlates greater than 0.95, then we're pretty happy. Obviously, we were adding noise to generate the synthetic data, so it's never going to correlate 0.1. But here, this at least shows us that we're pretty much getting everything right. Okay, so in conclusion, I doubt I needed to convince you. The fact that you're coming to this group means you probably already bought into the idea that we have a reproducibility problem in neuroimaging. I don't think it's any different from any other areas of science that have complex computational workflows in large data sets. And this is actually, what I've been hearing from people in other areas after the NARPS paper. I think that, we hope that data sharing can help overcome the problem of small samples, but it still remains a challenge. And we also hope that, that everybody in our field can start taking advantage of software engineering tools that can help us ensure reproducibility of our analyses, because there's a lot of tools that can help us do better science. Finally, I just want to thank the members of my lab shown here, people who have funded this work, and then a bunch of collaborators, but particularly Tom Schoenberg and Rotem Botvinnik-Nazaire from Tel Aviv University, who played a really important role in doing this work. So thank you, and I'm happy to take questions now.
Thank you, Russ. It was eye-opening and scary at the same time, to be honest. But thank you. That was a great talk. We have a question from Cyril, who says, "Hi, Russ. How do you plan to combine results from multiple pipelines? And would you differentiate processing path versus same processing but different models?"
Yeah, both good questions. Hi, Cyril. So we came up with an initial scheme working with Tom Nichols and Jeanette Mumford in the NARPS paper, which basically is kind of like doing meta-analysis across the workflows. There's been a lot of work in in statistics around things like specification analysis, vibration analysis, and actually thinking about taking advantage of some of those tools. But in terms of like the consensus, I think the main idea is to really kind of meta-analyze across workflows. So then the second question was about kind of pre-processing versus modeling, and the NARPS result. So we didn't actually see we didn't have enough different workflows to really analyze kind of which pre-processing choices make sense. The one thing that we saw that made us think that pre-processing might not be the big thing is that there was no different. We had about half the teams. We gave out, in addition to the raw data, we gave out data that had been pre-processed using fMRI prep, and about half the teams used that. The other half of the teams used their own results, and we didn't see any differences in the pattern of results between people who used fMRI prep versus people who didn't. And that, one takeaway from that is that, we think that it's pre-processing may not be such a big deal. The other thing that makes me think that it's really about the modeling is one, what we saw in the heat maps that I showed, right, where a big differences between teams had to do with, whether they mismodeled, whether they misspecified the model. But also, you see that across lots of different teams who have similar Z-stat maps, they still gave us differences in, kind of the hypothesis outcomes. This says that it's not just how you build the model, but also what you do with the map after modeling in terms of either ROI specification or thresholding. And so our initial work is really going to focus more on kind of what happens after pre-processing, in part because, it's a lot easier pre-processing is a much bigger computational load than modeling in general. And so it's more feasible to try a lot of different models than it is to try a lot of different pre-processing streams.
Great, thank you. I have a few questions myself because I've been working with fMRI for about 10 years now. And it strikes me that, like you said, ECRs don't have the opportunity to do exactly what they want to do with the data. But my main problem is we need to train our PIs. Usually you have a PI that says, I have a question, I have a technique, and I have a population, here you go. And then you end up with an ECR that has a year or 2 years to do a postdoc and publish very fast. So then everything that you say about, let's say, error hacking or software testing and all of this becomes very complicated. So that's something that I've been trying to do back in Toronto, trying to teach PIs what you should be thinking about before you even write a grant, so that you have enough time, enough money, and everything to actually, do all these things and make sure you thought about this beforehand. Otherwise, you're going to end up with a problem, if not a massive issue, with money that you cannot actually, or data that you cannot really use. So how do you think we can involve our PIs in this discussion? Because ECRs are always seeking training. PIs not so much.
Yep. I think that is the biggest challenge, right? And not it's a challenge at multiple levels, right? It's a challenge in the sense of like how they envision doing the science, because you're right that often people don't think about, doing things at a pace that will allow you to actually be reproducible. I think it's also a challenge around incentives, right? Because obviously all of this, if it were the case that, you got jobs in the end based on the quality of your work and not just on, what glossy magazines you published a paper in, then this would change overnight, right? But the problem is that it's people my age and above who are making those decisions about who gets hired or who gets tenure. And most of those people have not, I'm an outlier, right? Most people in my cohort have not, I think they're buying in slowly, but most of them have not bought into, this set of beliefs about, what the incentive structure should look like. So all that is to say, I agree that that's the hardest problem. And, that's part of why I've spent 10 years ago when I went to give talks, they were talks about like, hey, look at this, fancy new result we have. For the last 4 or 5 years, when I go give talks, they're exactly like the talk I gave today, which is, hey, look, we've got a serious problem and we need to fix things. And that's what I can do. I mean, that's one of the things I can do, right? The other is to make sure that at least in the decisions that I'm involved in, be as loud as I can about the need to worry about this stuff. And I know, again, that, certainly at Stanford, there's several of us in our department who care a lot about this. We started putting into our announcements that, evidence of open science and reproducibility practices, is appreciated in your, in one's applications. So I think another thing that you can think about is, really highlighting, like in one's CV, all of your open science practices. I just developed this new project called Auto CV that tries to help do that, where basically it puts in links to open code, open data, open access publications, all that sort of stuff. It's on my GitHub if you want to have a look at it. But I think that, anything you can do to kind of surface the fact that, people who are applying for jobs now care about this stuff and it's important enough to like end up on your CV. I think, that sort of signaling it's not going to make a huge difference, right? But every little thing that you and I can do to kind of highlight to, the gray hairs that, or the no hairs that, that this stuff is important. I think that, that's the job that we have. And unfortunately, there's no easy answer to this, right? It's compared to the technical stuff, this social engineering stuff is really hard.
Yes, I agree. Yes, it's a challenge. And even if you do agree with something with your PI, you often have to go back and, show emails and agreements in writing that were made a long time ago, because maybe the results are not satisfying anymore or enough. We have a few more questions. Would you say that fMRI studies are amendable to the register reports format? I ask because I am told that fMRI research presents unique problems at stage 1 review, given the many different ways to process and analyze the data. Any resources, templates, or advice would be appreciated. I've been involved in one registered report for an fMRI study, so that's a proof of concept that it can happen. And I don't know which journals are now doing. I'm pretty sure, actually, so this was a neuroimage one, and I'm not sure if it was a special issue or if they're generally accepting them. It is definitely challenging, right? Especially around kind of the data processing side. I think that, to the degree that one can actually simulate data and show, like I suggested, simulate data and show that, hey, look, we have this workflow. These are data like what we expect. We can actually, we can show here that if we put the data through our workflow, it has the error characteristics and the signal detection characteristics that we think it should. That would be really powerful. And we didn't do that in this particular one that I was involved in. We just said we're going to use this particular pipeline that we've always used. But I think it's certainly doable, right? And it really just comes down to being as specific as one can. I think in the context of fMRI, I worry a little bit less about, as long as somebody says, yeah, we're going to use this specific pre-processing workflow, I worry a little bit less about,
the specifics of that workflow. I worry a lot about what they say about exactly how they're going to define, the hypothesis being positive or negative, right? How are they going to define where the activation is, what it means for it to be, significant or activated, and so on.
Brilliant. I think there is also something that we need to be very honest about, something that came up recently in our team, where we pre-registered a pipeline and we figured that actually one of the software got updated after we've done our pre-registration, and we were not comfortable anymore using it. So I think if you're open with it and you say, yes, this was the best way to process the data at the time we wrote the pre-registration, but since the update, not so sure, then it's fine. I think you need to have this kind of room and be able to say, we tried our best and we figured that what we thought was best 2 months ago, even 2 weeks ago, is actually not that good anymore.
Yeah, we've never, every paper that we have published that had a pre-registered associated with it, a pre-registration associated with it, has ended up deviating in one way or another, right? And as long as you're very clear about what those deviations are and why you made them, obviously the reviewers have to judge that, but I think as a reviewer, I don't have a problem with that.
Yes. So we have someone that says, "Hi Russell, thank you for a fantastic talk. Do you think there could eventually be such a thing as too much replication, where fields move more towards focusing on validating previous studies and no longer explore new avenues?"
Yes, I think that's a serious concern. I mean, let me phrase it a little more generally, right? And I think I have a concern that, when people like me, talk about all of these things that one should do to do good science, that that listeners could walk away thinking, all I have to do is follow all those rules and I will be a good scientist, right? And unfortunately, that's not true, right? I think that being a good scientist involves doing good, reproducible, valid science, but also having interesting ideas. Yeah, because if replication is clearly essential, right? If we're going to build on results, I've had students spend several years in my lab as grad students, ultimately realizing that the one published finding that they were trying to build everything on doesn't replicate, right? And so they spent years that spent years of their life with nothing to show for it, which, under the current incentive structure is wasted. So I think that, one has to keep in mind that science is about understanding the world, right? And everything that I'm talking about is basically trying to make sure that when we say we have an answer about the world, right? And neuroimaging studies aren't going to change what people do in practice. Studies of COVID-19 vaccines will, right? And so it's but I think that across science we need to make sure that whatever we're doing, when we say there's an answer, that it's reproducible and reliable. So for example, I was just involved in writing a brief that has been submitted to the U.S. Supreme Court around somebody who's up for the death penalty, basically trying to argue that this person was 18 when they were sentenced to death in Texas for committing murder, based mostly on kind of somebody who didn't know what they were talking about, who predicted that this person is necessarily going to go on to be a criminal for the rest of their life. And basically, we use neuroscience in part to make the argument that, look, at 18, the brain is just not fully formed yet, so you can't predict what somebody is going to do later in life based on what they do at 18. And in writing that brief, there were a number of findings that people had initially kind of brought up that I wasn't comfortable including because they were such small samples that I didn't think they were reliable. And that's the place where this stuff becomes really important, right? If you're going to put something up in front of the Supreme Court, you want to make sure that it's actually reproducible. And so we certainly need reproducibility, but we also need people pushing the envelope in terms of new ideas.
Definitely. And we have Toby that is asking, so first he says, thanks for the talk. With shared data set, is there a risk of hitting false positive when too many people analyze them?
Yeah, we just had a paper in eLife last month, I think, by William Thompson, called Dataset Decay, which is about exactly this problem. What happens when everybody is working on the same data sets? And I think it is a, as somebody who does a lot of work sharing data, it pained me to have to write that paper, but once it became clear to us thinking about it, I think it's a serious problem, and I think we as a field need to think about how we're going to deal with it, and we're just starting to think about like what solutions might be, but it's certainly a problem.
Cool, thank you. So another question is, hi Russell, thank you for the great talk. You mentioned software engineering training. As many researchers are from different fields, can you talk a bit more about what is enough training or to write trusted code? How could we improve here? I sometimes really doubt if I am making bugs in my code, especially when using some new analysis methods.
Yeah, I mean, it's a really hard question to answer, right? I mean, there's, everybody's always learning, right? We're all always getting better, and it's a craft, right? Writing software is a skill and a craft. And so you never stop learning and getting better. One thing you can do, I think, is, work with others, especially if you have somebody around you who's more skilled than you are. But even if not, just like so there's this thing people call pair programming, where you basically write code as a pair, and you can do this, either together in the same room or using some of these collaborative things like Google Colab, where you can basically both kind of see the code at the same time. And that really kind of, drives you to have to think through what you're doing. And two novices are much better than one novice, right? Because they're going to see different things, they're going to make different mistakes, and you can really learn together. I think the second thing is like just finding the tools that are useful for whatever kind of work, whatever language you work in. Python. So I code in Microsoft or in VS Code, Visual Studio Code, and there's tools to automatically, like every time you save it, it does the code quality analysis. There's also things that can tell you, do sort of what's called refactoring, to basically say, hey, look, you're doing this thing, but it's kind of clunky. You should do it in this better way, and it can sort of automatically fix it for you. That's cool because it automatically fixes it, but it also kind of tells you, hey, look, these are the things you should be looking for. So any sort of automated tools you can find, I think, are really useful. And then finally, just finding relevant books and reading them. One that I'm reading a book right now by Martin Fowler called Refactoring, which is basically all about how to take your code and improve it once it's written. And it goes through basically a bunch of like generic sort of language. He writes it in the context of a particular language. I think he's using JavaScript, but the ideas are really generic, right? Like, you should, like, if you're going to if you're going to use a variable in two places, you should define it as close as you can to where you're going to next use it, things like that. And so there's a lot of like strategies from software engineering that I think one can find. Another great book I would recommend is called Code Complete. It's relatively old, but I think it's like 10 years old or so, but it's a really good book for kind of laying out like all the stuff you should care about. And I think really the last thing I would say is like really learn how to test your software. Do things like, put in answers that you put in data where you know what the answer is going to be, and make sure it gives you the answer back that you expect.
Brilliant. I think also one thing that helped me when I started with fMRI was getting a membership in all these forums about archives of SPM, FSL, and all this, just because then people talk about all these issues and it can make you realize that you're doing something wrong yourself. So I think it's also communicating with a lot of people. If you know that there's a postdoc somewhere that has more experience than you, just go ask. And if it's online on a forum, don't hesitate, because then you will have a lot more insight and your work will be a lot more productive and efficient and errorless, hopefully.
Indeed. Well, one thing to add, if you're not familiar with neurostars.org, which is a great forum for lots of different software for neuroimaging, that's a place to check out.
Definitely. So another question is, a previous speaker, Tal Yarkoni, discussed the generalizability crisis. He says that one possible solution, amongst others, is to have a constraints of generalizability section in the discussion. Do you think this is an initiative that neuroimaging journals could get behind, given that the undue generalization is arguably an issue in neuroimaging? I'm thinking particularly in regard issues with reverse inference in cognitive neuroscience.
Yeah, no, I mean, I love Tal's paper and his work on that, and I think in general, journals need to push people more to not oversell their results and to really highlight the limits on generalizability. So I would strongly, if I was editing a neuroimaging journal right now, I would definitely push for something like that to become a standard part of what reviewers look for, because I think the problems for us are much bigger than the problems that he lays out in his paper, right? Because we have all the problems he laid out in terms of generalizability across stimuli, across models, stuff like that. But then we also have generalizability across all the analytic choices, right? And then and obviously the populations as well. So I think it's really important.
Brilliant. We have the last two questions. I'm just going to close the Q&A because we're already a bit over time. So is there any way we can introduce red teams, an initiative proposed recently by Daniel Lakens? In other words, could money be put aside for bug bounties?
Sorry, for bug bounties?
Bounties, yes.
Interesting. I'm not familiar with that paper. That's an interesting idea. Yeah, I mean, my worry about that, I mean, I've always been on the fence about this kind of like, what do they call themselves? Data thug sort of thing, right? People who just spend all their time going out trying to tear other people's work apart. And I worry that kind of scheme sort of, it just builds a bad dynamic, a dynamic of like people going after other people. I think that, we need to be working together, right? And not making it seem like a kind of like a good people versus bad people thing, or like a good scientist versus bad scientist thing. That it's really, I think the large majority really just want to do the right thing, and it's really about knowing how to do it and being able to be in a position to do it and still meet the incentive structures. So I like the idea of putting resources behind testing and finding potential problems. And so, for example, if one could say we're going to put money into our grant to hire someone to come in and review what we've done and make sure that, if it's like an internal bug finding thing, I love that, right? If it's an external like data thug support thing, not to say that the work that's been done by, some people like James Heathers isn't important. I think it is. I just worry about the dynamic that it sets up.
No, definitely. I think it's something that has to do with the culture of research, and we're already trying to change this to make sure that we have less pressure, less bullying and harassment, less everything, less fear in publishing. So if you do induce this external pressure, you're never going to publish anything. This is going to be very complicated, at least.
Yeah.
So the final question is, registering clinical trial has been a massive help to constraining imaginative ways to analyze data. However, we've also learned that it is not routine to compare registered protocol and analysis with the final published article. When people have done this comparison, we still find issues with misreporting and selective reporting. What can we do to put more lead in the pre-registration pencil?
Yeah, I mean, I think this is largely a review issue, right? That reviewers need to look more closely. So there's just this recent thing about this paper in Psychological Science that was retracted on was it on like violence across countries and IQ, and it turns out that they said they had pre-registered it, and then when somebody went and looked, I think that was Nick Brown that wrote about this, somebody went and actually looked, and the pre-registration was written and submitted like 18 months after the paper had been initially submitted. And so, a reviewer should have caught that, right? Or I guess in that case it was the editor, because it was added, later in the process in revision. So I think that this is really just on the, that editors and reviewers, when something is pre-registered, need to be looking at it and checking it to make sure that it's legit.
Definitely, and check the different version of the registration as well, because you can always update it.
Yeah.
Well, I still have a million other questions, but I think it's time for everybody to relax in their evening, because it's 7 p.m. here. So thank you very much for attending. It was a lovely, lovely talk, and hopefully this will give us a lot of solutions for the future in neuroimaging. Thank you, Russ. Thank you, everybody. See you next week for Olivia Guest. Bye.
All right. Thanks, everybody. Bye-bye.